I'm Laura Bonnell, and this is the Living with Cystic Fibrosis podcast coming to you from Detroit. Please subscribe and even rate and comment on our podcasts when it moves you. I learn something new with every podcast, and I'm always inspired by the people that are showcased here. We don't give medical advice. You need to connect with your doctor for that. I hope that this Living with Cystic Fibrosis podcast educates you and sparks some volunteering or advocacy. The CF community needs people like you. Thanks to our sponsor, Viatris, for their support. A new CF modulator could be on the horizon. Professor Dr. Bhanu Jena is excited to tell us all about it. Dr. Jena was born in a small town in Odisha, India. He got his love for science and medicine from his father and grandfather. He majored in chemistry, zoology, and botany at BJB College in India, and he got his master's in zoology also. He received the Peasant Ku Memorial Prize and another gold medal prize as well. He also got his doctorate in zoology in molecular endocrinology, and his resume is a lengthy and impressive. He was a fellow at Yale University of Medicine and worked as faculty there. He also worked in the Department of Physiology at Wayne State University. And Dr. Jena was conferred the title of Distinguished Professor and George E. Palade University Professorship by the Board of Governors at Wayne State. And this is just a small introduction to who he is because, again, his resume is so long. Dr. Jena is a cell biologist and chemist. He unraveled the understanding of cell secretion with his pioneering discovery of the porosome, the secretion machinery in cells. And he is going to talk about this and explain it so that we all understand. Welcome. Thank you so much, Dr. Jena, for joining us on this podcast. So many exciting things to talk about that you're working on. So welcome.
Thank you.
First thing, let's just kind of start with the basics of what you're doing so that people can know. Can you explain in simple terms what porosome, what that means?
Yes. We are made up of about 38 billion cells. And these cells, they communicate with each other. And these communications are due to chemicals that are secreted. For example, nerves talk to other nerves, and neurotransmitters are secreted so they can talk with each other. And similarly, when we have a meal, our exocrine pancreas secrete digestive enzymes to digest the food, and that is through secretion. Once the food is digested, our blood sugar level goes up, and the beta cells secrete insulin to metabolize the sugar, the glucose. Similarly, other cells, such as, for example, in the lungs, secrete mucus. And this mucus, in the right consistency, is able to travel through cilia that beats in the lung epithelia and remove the mucus with its associated particles that we breathe in, such as bacteria, dust particles, and so on and so forth, and keeps the lung clean. So all these processes are actually managed through a mechanism in cells called cell secretion. And the secretory material is packaged in vesicles, in membranous vesicles, that dock and fuse at the plasma membrane structure called the porosome. And these structures are made up of 30 proteins. They are cup-shaped structures, 30 plus proteins, cup-shaped structures that range in size in the neurons from 15 nanometers to about 100 nanometers in diameter. And these structures allow these secretory material to be released from the cell. So they are very important portals in the cell that keep homeostasis in an organism. And any defects in these proteins or in the structure that can result from mutations that happens in cystic fibrosis, because one of the protein in cystic fibrosis is actually the CFTR protein. It's a chloride channel, which is one of the 30 plus proteins in the porosome complex. And that will make the secretion of mucus defective. And so that's what we discovered about 30 years ago, the porosome structure. We know the chemistry. We have reconstituted the porosome functionally into lipid membranes, into artificial lipid membranes. We have also reconstituted them into live cells for therapeutic applications. So a good example is, say, in diabetes, insulin secretion is compromised, and we can reconstitute the porosome complex that allow normal functioning of secretion of insulin from beta cells. And similarly, we have achieved reconstitution of the porosome into human epithelial, airways epithelial cells that allows them to release or secrete normal mucus and therefore overcome the disease.
And I think that explanation is really helpful. And I like the video that you sent me, and I put the link of that very short video in our show notes so that people can watch it, because it beautifully demonstrates exactly what you're talking about, but you can visually see it. So it makes it easier for you to kind of understand exactly what you just described to us. So that was really helpful. Tell us, you started this company and what you're trying to do for the CF community. I know it'll impact other diseases as well, but what are you trying to do with porosome therapeutics?
So that's a good question. I'm a scientist through and through all my life.
You are. And boy, what you have done just on paper and talking to you, because I've had a chance to talk with you a little bit before this. You are so smart. I'm so grateful that you are on this earth. Like, wow. It's just talking to you. I learn something constantly. And anyways, I had to interrupt because you're like a genius to me. And your vision for the future is just so refreshing and wonderful. But go ahead and continue with your answer. But I just had to say that.
Laura, you're so kind. But let me tell you this. So for many years, we're trying to understand how cells secrete and mechanism that is involved. And we're lucky that we use the right tools and technologies available almost 35 years ago. And we found this structure. And we found that it is a beautiful structure in the cells that allows cell secretion, which is a fundamental aspect of cell physiology. And we did all the work to understand this and its mechanism and how it works. But I never thought I'll start a company. The reason for that is very simple. As a scientist, you want to you're excited finding new things, how life works. But you never think about how. So I was giving a talk many years ago, actually in 2014, to students, undergraduate students who invited me to Harvard to give a talk. And some of these kids who are so bright, you said, I'm bright, I think they're brighter kids. And they said, oh, if this can help, there are many diseases that are due to faulty secretory mechanism like Alzheimer's and diabetes. Have you thought about that? And I'll be honest with you, and that's when it struck me. And I thought for a moment, I said, well, if we do all this work, there are many beautiful work that is done that is not translated or to benefit society, and it stays in the notebooks. And I should do something. So I met these two gentlemen who have done very well in business. And in this discussion, they said, well, we'll help you start a company that will allow these findings to be translated to help disease. So during the pandemic, we actually found a mechanism by which we can prevent any type of virus, cold virus, to get into the lungs. And then I had a student who was working with me in cystic fibrosis. In one of these papers, he is listed. And I said, you know what? I think this is a good idea. I have to think more how to translate this. And so these two gentlemen actually helped me. They are, Gil Mermel and Roy Verghese and some other people that funded the science and the company. And I am more excited that the work that we did for almost 45 years will help people with certain ailments. And for me, that is the most fulfilling part of having a company. And people talk about investments, this, that. I'm not into those kind of things. But now I realize that if it can help, that is my reward actually for the science that we do. And so I was excited when we found that in cystic fibrosis, there are 2,000 plus mutations.
Mm-hmm.
And the 508 mutation is the most common mutation, but there are also other forms of phenotype cystic CF disease where there are different mutations. There are also phenotypes where CFTR, the chloride channel, which is defective in CF, is not even present. And so it occurred to me, I said, wait a minute, if we can, this is a complex structure, which is 30 plus proteins. And for many years, I have talked to several experts in the field. They said, we have tried gene therapies and other things. It is not working. I said, I think the reason could be because we have this structure that we know that is made of 34 proteins. And if one of the protein is defective, it is very difficult to replace that protein in that structure because already the faulty, defective protein is there. And then it turned out that when we do a mass spec of the porosome complex in this defective CF mutated CFTR, the long epithelial cells from humans, we found that not only the CFTR protein has that mutation, but it disallows other normal porosome proteins to associate it properly with that complex to form the active secretory machinery. And therefore, now we understand the disease much more clearly, which means that the entire structure that is releasing the mucus has to be corrected. And that's the reason why we turn to reconstitution and let's see what it does. And to our surprise, it turned out that all type of mutations now that have the CF phenotype can be corrected by reconstituting the porosome complex. So we are very happy actually that it can help people of all different types, so we don't have to select individuals for any type of clinical, to do all the clinical trials. Everyone is welcome to join into the trial because it can overcome the CF for any type of mutations or even people that don't have the CFTR protein cells. So I'm very happy because of that more than anything.
Right. And it's very exciting because this means this would not only help the 10%, it would be able to impact the entire CF community potentially, correct?
Yes. So usually the 508 mutation, as you may know, because that's about 70% of the CFTR patients, 70, 75. And then other mutations are there. And you are correct. So now we are able to treat everyone. And that's the joy of it. Because if we can treat everyone, then all patients are taken care of and get this therapy. And so that's the reason why we're so excited.
And then you're just in the beginning stages of this, correct?
Yeah, so we have done organoid studies and we're doing animal studies. But I think more importantly, what this is also going to do is, and we know this is going to happen, so similar studies that was done for Trikafta, that is Vertex Pharmaceutical. It's a beautiful drug that is now helping a lot of people. And it's a very powerful drug, which is helping. And so what we think it'll do is we have now focused the lung. And the lung, the reason we focus the lung is, number one, it's easier for us because it can be taken as an aerosol through what CF patients are now using for their treatments. But also, so the lung is the most important, which actually dictates the morbidity and mortality in CF patients. But other organs such as digestive, reproductive organs, the liver is impacted. Those can for now, also be taken care by the Trikafta drug. So a combinatorial Trikafta drug is going to work. But remember this, or this Trikafta has only limited the 10% that is not affected can be dealt with by the Trikafta drug, but the reconstitution can. And so if we do it combinatorially, it is going to be very, very helpful. It's going to also decrease the amount of drug that is required, the frequency of the Trikafta drug that would be required. And I have a feeling we have now data that supports where we can take once the reconstitution therapy, and you don't have to take it for a week, but I have a feeling that it may be months before you have to take once, maybe in 2 months. And that's, you don't have to go to a physician. You can just take it yourself. It's in a little vial. You can just use your aerosol and take it. And these are very small particles. They are nanomachines, and they are present in all of us. So it's not going to have an immune response. So it has many, many positive impact on the health and well-being of the patient. We are very, very excited to move this forward. We also are welcoming this, other large pharma groups to participate with us, because it's at the end of the day, we are helping the patient, and we are very happy to work with them together to make this possible, get this therapy as soon as possible to all CF patients.
And just to clarify a little bit, so it would be an aerosol. It was something that, how many years out do you think it is before it can go before the FDA for, approval? We're talking years?
I think because we have now a large scale, we are able to scale it up very quickly. We know exactly the technology to do that. We have the resources to do that. It can be within a year. We are writing a grant. We're submitting a grant to the FDA for making the platform more helpful for designing and developing not only CF drugs, but many drugs. So let me give you an example. So as we find that in CF patients, say 508 mutation, there are 10 other proteins that is impacted by that mutation in the secretory portal, in the machinery. Now, what we are also doing is having small molecule drugs impacting all of those proteins, so that not only the CFTR protein, but the entire machinery is now functioning properly. So that's another thing that we're doing. So those will take time, but the reconstitution therapy can immediately come to people within a year. If we get support from the Cystic Fibrosis Foundation, we get support from the FDA, and we can move forward with that very, very rapidly. And I think most importantly, that would be a therapy that would be an immediate pathway to a cure that the CF Foundation is looking for.
That's a huge thought and promise. That's huge.
I'll tell you, Laura, we are very excited because at least in the organoid level, we are more than twice as effective as the Trikafta drug. But again, I say again and again that Trikafta has been one of the best medications for CFTR in helping a lot of people. But always there is space for development and better therapeutic applications that throughout history, the medicine community has focused on. And so this is one of them. And so we're happy Trikafta started, I think, in 2019. People were started to give, and it has helped a lot of people. Now we have this therapy that can be complementary given with Trikafta that is going to better help and support the patient community suffering from or impacted by the CF. But I think more importantly, all CF patients can have it. That's, I think, the important message that I want to tell.
The dream, that's the hope that everybody would have access. But I just wanted to clarify one thing that, we've been talking about that you said was your inhaled medication would help the lungs and it would work together with Trikafta or possibly something else if something else comes out in the future, because yours wouldn't work completely alone. Is that correct?
Well, it'll work completely alone in the lungs. So because the reconstitution is being done in the lungs. And that's why the FDA grant is going on. So we want to reconstitute them so for example, in the liver, the bile secretory passage is blocked by mucus, and therefore the bile is not secreted, and therefore it has a lot of negative effect on the liver, and the liver is impacted. A lot of scar tissues, and a lot of inflammatory. And similarly for reproductive organs and for digestive, and so the pancreas and so on and so forth. Those therapeutically to reconstitute, we have not done the optimal studies how we could do that. But the lung, we're lucky that the lung not only is one of the most important organs that is impacted by CF, but we are able to treat it. And it is much easier because all you do is inhale it, and the small little particles that go all over the place, we need very little of it, and it helps the lung release the normal mucus, secrete the normal mucus, and therefore abrogate, bad effects of the mutated CFTR and thick mucus that is secreted.
Have you talked to other scientists like, I don't know, Dr. Francis Collins or Dr. Mitch Drum or any of the guys or gals that have already worked on CF? I mean, I'm just curious to see what their response would be to you, or if you've talked to any of them.
First, let me say this. In our company, our chairman of the scientific advisory board, Professor Joachim Frank, he won his Nobel Prize in 2017. He's a professor in Columbia, and he is personally working with colleagues at Columbia on the structure of the chloride channel on CFTR. So he's the number one. And then we are in collaboration with the Cystic Fibrosis Center, Animal Center, which is the largest in the world for CF type of work in Case Western. We're also working with Doug Tages, who is a long-term collaborator with me when I was at Yale. I started my career there. And Doug and I have been working for almost 30, 35 years or more on many different aspects. And he's a CFTR and just published a paper recently. And I think with, I try to remember his name, I'm forgetting, who actually won an award from the CF Foundation as a Leaders Award or some Pioneer Award from the CF Foundation. So yes, in other words, and I was invited many years ago to the Garden Conference for CF once we found that the CFTR protein in the lung epithelia is associated with the porosome complex. And so I presented there and met a lot of wonderful people that have done lifelong work on CF and learned a lot. And that was my initiation into the CF world in 2015 at the Garden Conference in Texas, I think.
It's very exciting. I know it's hard to recollect everything and every paper everyone's done, but I think it's impressive and it's wonderful. And I'm also excited. You're not just in Michigan, but you are in my home state of Michigan. And we haven't met in person yet, but we're going to, because we're only like 15 minutes away from each other. And you're a professor at Wayne State University, among the other places, which I mentioned in the introduction. But it's exciting to have you here in Michigan doing great things.
Thank you, Laura. Thank you. And so is your foundation that is doing outstanding work for so many years. When we first met in our friends and family of CF, which I'm very close to now with so many people that I interact, including the Institute in California and discussing with them.
CFRI with Siri Vaith.
Yes.
Yep. Love her, our girl.
Yes. And also the scientists that are involved who are clinicians. And I think he's one of the leading scientists that did a lot of work on genetic studies on CF and from Stanford University Medical School, who actually said that for many, many years, he has been trying therapeutic through a gene therapy and it has not been successful. And that's when I told him, this is why it has not been working, because we're just focused on one now protein. However, when mutations happen, the porosome is completely disorganized, and therefore it had to be treated in a systems level approach. Let's put it that way. So I'm very happy to know these people, but I'm also humbled because 10 years ago, I never expected where I'll be in terms of helping the CF, the disease, because I'm not an expert in CF and slowly learned over the years, but very grateful to all the science that has been done, the background science, and that's why we're able to do what we are doing. So all the work that you guys have done, the Institute, the CF Foundation, all the scientists that have been involved in the CF community doing beautiful work they've done, allows us to be on this platform and understand the disease properly, whereby we are now able to utilize the technology that we have developed to help. Without that background, without those science that was done by all these scientists, I would not be here. So I'm grateful for that, Laura.
And all great points. And thanks for your kind words as well. But yeah, I'm glad you found the CF community, or it found you, however it happened. I'm glad you're on board. What are some of the big things that you're trying to change in the CF community, and how is your work going to impact, CF around the world?
Well, there are other therapeutic applications that we are trying. So, for example, we are trying the gene therapy and the mRNA therapy that will now target all those porosome proteins that are impacted by the mutations on the CF. And therefore, when we raise them, now the technology is there whereby we can use the mRNA, multiple mRNAs, to upregulate proteins that are being impacted by the CFTR protein mutation in the porosome complex. So that's one we're doing. Second, what we are doing is screening small molecules that will now not only impact the work of the Trikafta is doing, but it is also not addressing the other proteins that have lost the capabilities for secreting properly the mucus. And if we target those, then we may be able to move that forward, that therapeutic. That means Trikafta, along with all these other small molecules, will impact and maybe have 100% impact on returning the patient to normal mucus secretion, just like the reconstitution is doing. So we are working on those type of things. And then, as I told you earlier, I think the reconstitution, we're also looking for impacting other tissues like the liver, the digestive organs that are impacted, the reproductive organs that are impacted. So we're working on those, both reconstitution and small molecules. So that's where we are. So it's a long road, but at least the light is there that will lead us, because we have a therapy and for everyone. And that's my excitement. And if it is there, if it has little to no side effects because it's a nano machine, which is in all of us, there's no immune reaction for that. And therefore, it is going to impact very, very positively to overcome CF. So that's what I'm all excited. So I'm focused actually on the reconstitution therapy, and I want to get this out ASAP to the patients. And for me, that is the goal I have for 2024. I'm hoping we'll be able to make it.
Right. So are we. So are all those patients. Absolutely. Cheering you on and waiting. Definitely. There's a lot of hope in what you're doing, and a lot of people waiting for you to say, it made it through the FDA and that it's on the way. So there's got to be a lot of pressure for you also to make this happen, because people are waiting.
That's true. But one important thing as a scientist is, before we came on and said what we said, there's 30 plus years of science that is behind it. And I think usually as a scientist, you should be a self-critique. So I started off not as a business, but as a scientist, I'm never a businessman, but I think you have to look at the safety issue. that's for me as a scientist, that's more important. So before we come out and say it can therapeutically help, we actually made sure that there is a safety issue on this one. And that's what I'm happy about. There's a huge safety issue in this. We are developing, and I'm not allowed to say for the company, but we are developing certain other therapies too for CF, that is stem cell therapy, but in a different way that is out there now, which is taken from the patient and utilized. And we are moving fast on those two. However, this is something that is really, really safe and we can scale it up. And only then I came out and said, let's do it. So for me, hopefully, jointly with a company who has track record on this, we'll be able to partner and get this out to the public. I don't want to wait too long, because usually companies, they wait long, they want to do it themselves and make all the profits and so on and so forth. I told our CEO, I'm not interested in that. What is important is we have to get this out to the patients. And if we can do that with the help of large pharmaceutical company who have all the infrastructures and other things to take care of, besides the science, we can do it very quickly. And that's what I'm invested in doing. And, the same thing also happened with mRNA. mRNA vaccine, mRNA was developed almost 60 years ago in France by Honored and other people who identified the mRNA. It waited for 60 years to translate into healthcare. And if we didn't have the pandemic, it'd still wait for decades before it is used. But, lucky we had the technology, and so it helped to develop vaccines. And now it is also going to help other therapeutic aspects. So this is similar. So we had, we have done this for over 40 years, 30 years, the discovery. So most of the work is done. So we are very confident about our science. So now the question is how to translate it in terms of its commercialization, let's put it that way.
Mm-hmm.
And I'm not an expert. And so a pharma collaboration is important, I think.
Well, it's exciting. And I know you've reached out to other companies and, and you're welcoming that. So I hope it pays off, because it will definitely benefit the CF community. Thanks for telling us about it. Is there anything, as we're wrapping up this podcast, that you didn't mention that you wanted to say about what you're working on?
We are working on many other therapeutic approaches using the poison therapy. So that in CF patients and others, patients that are especially the older people, even younger people, when we have a problem and we're given the ventilator impacts the muscles of the diaphragm. And we found a therapy that is taken from the patient and can be given back to the patient in the diaphragm that will prevent atrophy of the diaphragm muscle and therefore allow the CF patients to breathe well.
And is that like gene therapy transfer? That's something.
Yeah, so we have a patent on that. But, and this new therapy now, it is going to help. Not only that, it's going to help people that are in the ICU.
Great.
For myopathy, it's going to help people that are now suffering from using these weight loss medications. They'll lose the fat, but the muscle will be protected. We'll be talking with NASA, because our astronauts and other people that are going into space will lose muscle mass rapidly, 6-fold faster than in the normal atmosphere. And therefore, our astronauts are protected. And so this is a huge, it is a separate spin-off company, but it's also going to help the CF patients too.
Well, it's very exciting. You have so much going on. And I thank you for everything you're doing and for sharing some of what you're doing with our audience. So thank you very much.
Thank you, Laura, for having me. And we should go get together and meet in person.
Absolutely. Very soon.
And let me know anything I can do for your foundation.
Thank you. The original music in this podcast is performed by Kevin Allen,
It's not complicated.
who happens to have cystic fibrosis.
We all got our worries and fears. I know it's got you frustrating, but loving you is so alright.
This has been the Living with Cystic Fibrosis podcast. For more information and to learn more about the Bonnell Foundation, visit our website at thebonnellfoundation.org. That's the BONNELL Foundation.org.
This podcast was produced by Jag and Detroit Podcasts. Follow our show on Apple, Spotify, YouTube, or wherever you're listening right now.